Good afternoon, respected Principal Sir, Chief Guest of this session, Dr. P. MALLIKARJUNA Sir, and faculty and students attending this FDP session. Welcome to the afternoon session of the 3 days FDP hands-on workshop on AI in drug discovery and development, a lung cancer case study. So I welcome you all to the afternoon session. Now I request our guest speaker, Dr. P. Mallikarjun Rao sir, to hand over this afternoon session. Sir, I welcome you to the session, sir.
PDB or 6 PDB. Okay. Okay.
Sir, what?
PDB.
అది లేదు సార్ సార్ అది లేదు
Sorry, what? RCFP PDB.
సార్ కి ఫోకస్ చేయండి.
Okay. Then go to the Campbell. Okay. Now, so far we have discussed about the genes. Okay. Then gene network and 3D structures of the proteins. Okay. And also we talked about how to collect the literature. Okay. How to collect the biological relevant literature from PubMed. Okay. Now we are moving to the next step where we are going to talk about the chemistry. Okay. So in drug discovery part, the first one is biology. Then we are going to talk about some chemistry. Okay. Now there is one database which is called as a Campbell. Okay. And this actually shows the association between the chemistry and the biology. Okay. That means here you talk more about actually the activities, okay, the potencies, and also you talk about the safety. Okay. Campbell is a manually curated database of bioactive molecules with drug-like properties. Okay. Actually, this is manually curated. What does it mean is, actually this one is a government, UK government initiative, which is society which is called as European Bioinformatics Institute, EBI. Okay. So what they do is all the papers that is available in the literature, okay, they will send those papers to the, they have employed few people, okay, and majority of those are from India, okay, and they work from home, okay, and they will ask the people, okay, to extract the chemical information, and also they ask them to extract the biological activity data, okay, and and they will curate it into a database. That's why it is called as it's a manually curated database of bioactive molecules. That means the molecules that show some kind of a beneficial pharmacological or biological activity, okay, with drug-like properties. It links compounds, targets, assays, activities, drug mechanisms, and clinical development information. That means it is going to tell everything. It is going to tell everything about the drug discovery process. That is the next phase of the drug discovery phase. Okay, that means uh, until now, whatever we discuss, it is about the biology of the targets. Now we are looking at the chemical information. Okay. In drug discovery, it helps answer what compounds have been tested. Okay, against which target they were tested, under what assay conditions, and how active are they. It will tell everything. And this one we are taking as EGFR inhibitors as a practical example. Okay. Then this is when Campbell as a description support tool, not only a data lookup website. Actually, we can do lots of things in Campbell. Okay, and it will interpret the IC50 value, Ka value, KD value, and the EC50 value. Okay, and also we are going to talk about what are the common mistakes that what we do. Okay, this one turn the literature in bioactivity into structured drug discovery evidence. Okay, actually those who are from the chemistry background, okay, and when they do this Campbell database, actually using Campbell database itself, we can write a lots of papers. We can write a lots of papers, and it's being updated so fast, so it is very difficult sometimes to catch up. Okay, but that I talk about it later. Next. Okay, now from known biology to the bioactivity informed compound decisions. That means the biology is the one that we talked in the past. Okay, gene codes, strings, PubMed, and AlphaFold. Now we are, of course, there is one more missing, that is PDB. That one I will check later. Okay, and to bioactivity informed compound decisions. Okay, this is the validated target. Okay, from Campbell we can get this one. Known ligands. Okay, now here you see a separate word, ligand. Ligand means any compound, any chemical compound that binds to a receptor or that binds to an enzyme producing a biological response is called as ligand. And ligand can be a drug or need not to be a drug. Okay, so that is the ligand, and we also will tell you the bioactivity data, and it will give you lots of insights on how the structural features of a particular chemical compound or a particular chemical is affecting the activity. Okay, that is structure activity relationship, and actually from that also we can check the candidate selection. Then later on we can go to the variability. Okay, so here target validation through known active compounds and clinical candidates. Okay, here the clinical candidates means the compounds which entered into the clinical trials, Okay, then of course from this actually we can identify the lead and we can modify those things. Okay, and we can also do the structure activity relationship analysis. Okay, and selectivity assessment by checking activity across target families. That means here, when I'm talking about the selectivity, and we talked about the normal EGFR and mutant EGFR. So what do you think? When you develop a molecule, do you think that it is going to work on both, or it should work both only on mutant and it should not work on wild type?
Only mutant.
Yeah, it should work only on mutant type, but not wild type. Okay, so that is called as the selectivity and the repurposing ideas. Okay, I think repurposing is. Okay, can you tell me actually how many medicines or how many small molecule medicines are available in the market? Over a rough guess. It's only between 2,200 to 2,400. All the medicines that are available, small molecule medicines, are only between 2,200 to 2,400. Okay. What the people will say that is, it is possible to synthesize a compounds of 10 to the power of 60 different combinations we can prepare, but out of which only so far what we know is only 0.01%. That's why the chemistry is still evolving. Okay? Now, repurposing means, for example, let us say we discussed about today morning, we talked about metformin. Metformin is known for the treatment of diabetes. At the same time, metformin can also be not used, can also reduce the weight. Okay, but however, it is not yet approved. So, repurposing means for you take a drug of known bioactivity, then you try to explore the new uses of the existing medicine. That is called repurposing. It is one of the strategies that the people use, okay, in drug discovery. So for EGFR mutated uh, non-small cell lung cancer, Campbell helped compare the activity of all these compounds. It supports decision of the reversible versus covalent inhibition. It also helps understand why potency must be interpreted together with the mechanism, mutation coverage, selectivity, and effect. Okay, that means whenever you say, because most of the time I also see that the people say that I am synthesizing a drug-like molecules. Okay, that's what they say. But when they do the activity, they do the activity only on biological target. From that, actually, we cannot say it's a drug-like. Okay, if you want to say drug-like, actually you should do all these tests. Mechanism should be established, and you need to see whether that molecule is also working on the mutated proteins or not, and it also should show the some selectivity profile and the advent properties. Okay, so known drugs are reference points for designing the better candidates. Okay, the next one. Okay, in Campbell, when you open the Campbell, you will have all these things: compound, target, assay, activity, document, and drug mechanism. Okay, so when you go to the compound, it will show you the chemical structure and what are the synonyms of that particular compound and what are the different identifiers. Okay, and also you will know the properties. Okay, of course, you can also check about the target. Okay, there you can talk about the EGFR. Then it will tell you what is the protein, what is the complex. Okay, and in which organism these are being expressed, and also the target classification it will tell. And this one, essay. Actually, if anyone wants to do a research and if they want to check what are the available experimental protocols to test the activity of that particular compound, actually, if you check here, am I right? Essay, it will tell you. You need not to check. So you need to see whatever the experimental model that you are doing, you see how many people already did in the past. Then only the people will, what is it called, will appreciate your results because you have used an essay model which is well accepted. Okay, in the scientific community. Also, you can also check the activity. Okay, then actually in that one. The activity of a compound, okay, it depends upon the type of experiment. You can explain, okay, before I go, can you tell me what is the difference between the potency and activity?
Activity is something it processes, and potency is the minimum quantity where it is sensitive.
Yeah. Activity means it produces a biological response. Okay, suppose there are 10 compounds, and if someone asks you, can you rank them? Which is the most active? Okay, so that one we have to look for a potency. Potency is the one that that is a something what we call it as. It will help you to rank the compounds in either ascending order or in the descending order. Okay, and whereas activity means it's a qualitative measure whether it is active or inactive. Okay, so the activity of a compound can be determined or can be represented either in IC50 value, which is inhibitory concentration, or Ki, that is called inhibitory constant. Okay, Kd, which is a dissociation constant, and EC50, effective concentration in 50, in 50% of the models. Okay, now can anyone tell what is meant by IC50?
Minimum inhibitor constant. Minimum inhibitor constant.
Okay, that means what happens is if you are testing a particular compound, okay, let us say in 100 models, okay, and it should show activity at least in a 50. At least in 50, it should show the activity. All right. So, but this is what all the standard textbook says. Okay. But in general, what we need to see is IC90 or IC90. That means IC90 the people use, the people also use 95. Suppose if you are talking about a cancer, so what we need to see that the concentration that is required to determine, to kill or to inhibit the growth of the cells. If it is by 50%, it is called IC50. If it is 60%, it is called as IC60. If it is 70%, you will call it as a IC70. Okay. So that means then you may wonder why the 50 became the standard marker. Why? Why in all the textbooks, why they say only 50, 50, 50, 50? The reason for that is if we invent a new medicine, we cannot expect that the new medicine is going to work for all the people, but at least it should benefit 50% of the population. Okay, with that concept, the original pharmacology was developed. Okay, suppose if any compound shows at least in a minimum of 50% of the population, then it will be considered as active. But nowadays, in a drug discovery project, always the people can go up to the 90, can go up to the 70, 80, depending upon the confidence levels. Okay. Of course, Ki and Kd is actually direct experiment. That means you take a receptor, to the receptor you will add a compound, then you will check the activity, then that is called as that, then the activity is determined by the Ki or Kd. Okay. Of course, you can also about the documents. That means you can get all the publications. You can get also the patents, everything you can get, and also you can also get the business. A chemical is the result of is meaningful only when compound, target, assay, and activity are interpreted together. So now you can see simple target to network to 3D structure. Now we are looking at chemistry and all this together. That means it is progressing into the more and more complex complexity of the drug discovery phase. Okay. Okay, this is the beginner question. Which compounds inhibit my target? Is my target druggable? What drugs or clinical candidates already exist? What is the reported IC50 value or Ka value? What is the Campbell ID for this compound or target? Because whenever you do this one, you need to note down the Campbell ID. Okay. Then, of course, when it comes to the expert, are the activity values comparable across systems? That's what the experienced researcher should look into. Okay, which compounds are potent, selective, and mechanistically possible. Okay, that's what we need to really check. Okay, and is the reported activity a biochemical activity, cellular, phenotypic, or binding phase? Okay, now you need to understand all these things. Okay, biochemical, cellular, phenotypic, or binding based. Biochemical means this is mainly applicable for the enzymes. Okay, so you all might have studied the biochemistry, am I right? And you can see a lots of chemistry is involved in the biological process. Okay, a biochemical assay is a simple assay method where when you are treating something with a compound, okay, with a chemical, you will see any kind of a response. Okay, so it is nothing but a simple chemical reaction. Okay, and to put it in a simple terms, am I right? When you are titrating if you want to estimate the concentration of the sodium hydroxide, what you do is you do the titrometry with HCl, and we know that HCl reacts with the sodium hydroxide, forms a sodium chloride. Then in that one you put a coloring reagent. So because of the pH, there will be a change in the color change, and you say that the color change says that the reaction has ended. From that you will go back and check and calculate the molarity of the experiment. Am I right? So in a similar way, in a biochemical also, what they do is they do some kind of a chemical reaction, and they will from that they will correlate back to the some bioactivity, whether it is active or not active. That is a biochemical assay. Okay, so the advantage of this biochemical assay is actually you don't require any living organism or you don't require any cell. Okay, but that's what actually the olden days that's where it was started. Okay, that is in our body, entire body, everything is a chemical. Everything is a chemical. Okay, even I'm talking about this one. So that is here. I'm making use of my glucose. Okay, that glucose nothing but a glycolysis cycle. When you look at the entire glycolysis cycle, you will understand that okay, how many ATPs are being consumed and how many ADPs are produced and all those things. Okay, so that is the biochemical one. Then the cellular assay means this is what most of the anti-cancer research people will do at the beginning. They take the cell and they put a compound, and from that they will try to see what is happening to that particular cell. So compared to the biochemical, because all these chemical reactions are happening within the cell, all right? So when you are doing the chemical reaction, yes, you know that it is active, but for your compound to be show the biological activity, we should go through the cell. Am I right? That's very important. If it does not go through the cell, then even though it is stopping a particular reaction, biochemical reaction, it is not useful. That's why the people think that the cellular assays are little bit better than this biochemical assay. Phenotypic assay means, okay, it's a general thing, okay. Just to imagine yourself, okay. In so when you are walking on the road, you will see lots of plants, but we eat only few plant leaves. Why and how? We don't eat all the leaves. Am I right? We eat only few leaves because our ancestors say that you eat these leaves. That is called as a phenotypic. Okay, so it is coming from this one or it come from the accidental. Okay, here also I can tell you one thing. There is a one plant which is called as a clerodendron. Okay, and from that actually one medicine, Ayurvedic medicine was developed. Thinking of it is for what is this called? It is used for the obesity. Okay, so you know how that was observed is, so in olden days there were not used to be compound walls, concrete walls. What they used to do is they used to put a plants, okay, around the house as a this one, and they observed that when they are putting a particular plant, these animals are not coming and eating the plant because that's what they want. Okay, when they put a plants, they want the animals should not eat that, am I right? Then when they see that one clerodendron, then the scientists think that why it is happening is because when they eat, actually they will become thin. Okay, yeah. From that only this one. And actually, if you see, animals are more intelligent, and of course, human being is also animal. And if you see the cows or buffaloes and all those things, If you see carefully, even the dogs and all those things also, they smell first, then only they eat. Okay, that means by sensing itself, they understand that is how the plant is, that is phenotypic. And of course, binding based assay is you take a receptor and you put the molecule and you see whether it is binding or not. Okay, does the data support a new program, repurposing idea, or structure activity relationship hypothesis, and what additional experiments are needed? Okay, better next slide. Now, if you know the target name, okay, then search for targets. Okay, here in this case, of course, if you know the compound name, then you put these things. Okay, if you know the Campbell ID, of course, when you are doing it, it's very difficult for you. Okay, and Campbell has these kind of IDs. Of course, if you know the chemical structure, you can draw the chemical structure into it. Okay, then you can find the analogs. Okay, of course, if you know the protein sequence, you can put the entire protein sequence, you can get it, and you can get a clinical drug class, and you can search the drug or mechanism types. Okay, do not search everything at once. Choose the route that matches the question. Okay, that's a very important point. Huh? Okay, this Campbell itself, okay, because this European Bioinformatics Institute, I worked, not I worked, I went few times. Okay, but the entire project is more than 500 million pounds project. Okay, that much money that they have spent on this. Okay, madam, next slide. Okay, search strategy one, that means target centric search. So first you need to put the target, then you have to see human target, then open the target phase, then the activities, then you filter the SS, then you interpret the data, search for the target name and confirm the correct organism. Prefer a specific single protein target and study the direct inhibition. Okay, and then you have to check the target type. Okay, then open activities and filter. Okay, all these things we can see later, and read essay descriptors. And this is the one of the this is the example that we are going to use EGFR. Filter the activity, check whether SS use wild type, detect cell base, and record activity value. And okay, next one. So this is one target eccentric search is best for building a ligand and activity landscape around a protein. So that means it depends upon you whether you are strong in chemistry, you put a chemical structure, or if you think that you are strong in biology, you put that biological data. Okay, the protein. Next one. Okay, now second one is compound centric search. Okay, then you can, if you know the name of the compound, you can put that. Okay, then you open the activity, see, you can record it. Okay, then same thing also you can get the same information. The next one.
Okay.
Next one. Okay, then potency values. Okay, here IC50 concentration giving a 50% inhibition, KI inhibition constant, KD binding dissociation constant, EC50 in a functional assay, then P Campbell. This is the standardized negative log activity value. Okay, and actually this one the people use a lot. Okay, which is called as PIC50. In all the computational models, they use this value. Okay, that's what it is said. Use it for modeling when standard relations is uh, when is appropriate. Okay, then a lower IC50 is not automatically a better drug. That's important. Lower IC50 means it may be active, very active, but it may not have uh, required safety or toxicity profile. Okay, next. So how to avoid being misled by a large activity table? Then you can put standard type IC50, KI, KD, or EC50. Then you can use this relation. You can choose the organism. You can choose the what is the type of assay that you want, and you look at the confidence score. Okay, and these are the results that you need to do it. Okay, next. Then from a tempting ranking to a valid comparison, okay, compare compounds within the same assay when possible. Okay, that means suppose if you put a EGFR, okay, there are many different experimental methods to analyze the or to assess the activity of the compound. So when you want to compare, you cannot put all of them together. So you need to identify the compounds based upon a particular assay. Okay, then you need to compare the structures, compare the activities. Okay, then use the same target construct, check if the endpoint is a biochemical, prefer standardized values, okay, and compare potency. Okay, and this is unsafe comparison, taking the lowest values from each compound across the unrelated assays, because the activity of the compound also depends upon the experimental model that you use. Okay, so when you want to compare the activities of the compound or relative potency of the compounds, you need to make sure that they use only one experimental protocol. Okay, that's important. Okay, comparing these things and ignoring ATP concentration in kinase assays. Okay, here just I want to tell you this one, because you know that ATP is converted to ADP. Okay, therefore it releases the energy and it causes the cancer cell to grow. If you put more ATP, it will move much faster. It will grow much faster. If you put less ATP, it will grow less. Okay. So especially in this kinase assays, we need to pay special attention on the ATP concentration. Okay. So when you want to compare the activities of the compound, you have to make sure that the ATP concentration in all those assays remains the same. Okay, that's the key point. Okay, and ignoring mutant versus wild type, yes, that also we need to check. And claiming a compound is best only from a potency alone, you cannot make it this one. Next one. Okay, and here you also can see this maximum phase. Okay, so can you tell me clinical trials how many phases are there? 4. What is 4?
Post marketing. Post marketing.
Post market. When it will end?
Till the drug is in the market.
Huh? It will not end, am I right? So yeah, that's why we say that drug discovery is a continuous process. Okay, and at least I'm happy to say that you say the 4 phases. Okay, actually many people say that it's only 3 phases. Okay, so that is the post-market surveillance, and which comes under the pharmacovigilance. Yeah, okay, that's very important. Okay, see here I want to say, because this is what really we emphasize there. Okay, of course, unfortunately here in most of the pharmacy shops, okay, we don't see a pharmacist. Okay, and if something happens also, actually there is no one who is reporting to the government. Okay, and actually in what is it called, in developed countries, okay, most of the time the medicines are dispensed by a pharmacist. Okay, and now in UK, actually there is called dispensing separation law. Dispensing separation law means the doctor only will diagnose what disease that a person has, and doctor won't tell what medicine that they should take. Okay, then with that information, it goes to a pharmacist. It goes to a pharmacist, and pharmacist is the one who says this one. Okay, and this is completely separation. Before this, because I'm telling this one because some of you are sleeping, that's why I feel that if I talk something which is not particularly scientific, it may be useful to you. That's why I'm telling. But it's okay. You can sleep. I can understand if I'm in your position. Don't feel sad, bad, or anything. Okay, if it is a power nap, it is good for you. Okay, yeah. So you, yeah. Before this one, what was happened was the doctor will give a medicine, am I right? And it goes to the pharmacist. Actually, pharmacist has to screen. Okay, that is called as whether the prescription is rational or not rational. And if the pharmacist feel that it is not rational, it will go back to the doctor, and the doctor has to change. Okay, so of course in US still it is being practiced in that way, whereas in UK they have completely separated, and Europe also they separated. Okay, but in some other countries still it is not happening. and that is the purpose of the Form D now, okay, and actually that is the main intention of introducing Form D, okay, into India, because at the time I was also in US together with the previous PCI president, when they are putting up proposal of the starting the Farm D programs in India. Okay, so but I'm not sure how effectively it is happening now. Okay, so but that is the purpose of the Farm D. The purpose of the Farm D is to help back to inform the government actually what is happening in the market after the particular medicine is uh, introduced into the market. Okay, so the maximum phase, a drug mechanism comments and indications, and first approval or regulatory report. So everything you will get it. And clinical maturity is useful context, but it does not replace mechanistic interpretation. Okay, so it will tell you a lot. When we look at all these things, we say, oh, Campbell is alone sufficient for me. I need not to do anything else. Okay, but I personally also more biased towards Campbell. Okay, because basically I'm a medicinal chemistry person. Okay, but still I advise the people to go look at all other databases, then only come to the Campbell. Okay, don't take shortcuts. Okay, next one. How Campbell helps selectivity thinking. Open a compound and review activity across multiple targets. Okay, look for activity of all these things and use target class. Okay, check whether activity is strong and remember that the absence of data is not evidence of selectivity. Okay, so when you are searching for a particular compound, you don't see any value. When you see you don't any value, it's not. It's not uh, it's not that it is not active. Okay, since I'm saying this one, just I want to tell you this one. For every experiment we put a positive control. What is the purpose of the positive control?
Comparative study.
Comparing our results.
Minimize the errors.
Actually, I was also told like that, but it's not true. Positive control is the one that determines whether your experiment is working properly or not, whether your experimental protocol is right or wrong. That is the purpose. Okay, when we say that, okay, so that's why selecting a positive control. But most of the time when I was studied also, I say that, oh, I'm designing a compound, I want to see that how potent my compound is compared to a positive control. Even that's what I was told, but actually it's not the truth. The truth is, because when you are performing an experiment, okay, you need to make sure that your experimental protocol is correct. How do you confirm whether your experimental protocol is correct or not? By using a positive control. Okay, and for each and every essay there is a separate positive control, and you need to use that positive control, not to compare the results. Actually, unfortunately, it's not. Okay, and of course, negative control. Everyone knows that whether it is because of the compound or it is because of this one, but many people will misinterpret in many ways of the negative control. Okay, so that one, okay. Check whether activity is strong, weak, or inferred. Okay, remember that. Okay, this one. Okay. So, easier for example, hepatitis shows broader family, and this one to better address this mutant. Selectivity must be interpreted, and Campbell can support selectivity hypothesis. Next one. Then you can also check the similarity search. Okay, so you can write something and you can look at what are the similar compounds that are reported in the literature. That also you can check using this molecule. Okay, using this particular search. Okay, so yeah, use when you have a known active compound and want related molecules, and especially this one because you know, since we are spending a lot of time and energy and also money in designing the molecules. Okay, we also want them to be protected by intellectual property rights, which we call it as a patents. All right. So that's why whenever we have some idea, okay, then we'll check in the literature whether the compound is reported in the literature or not. If the compound is reported in the literature, then what still you can do it, but you don't have it. You don't get any financial benefit. You don't get any financial benefit. Okay, can you tell me these industries and all those things? They spend a lot of money, am I right, on the drug discovery? How much it will cost? Huh? How much it will cost? Rough estimate in dollars.
500 million.
500 million US. It's starting from 1.5 to 2.5 billion US dollars, but they will get back. You know why there is a patent? Can you tell me what is the purpose of the patent?
Protection against money.
Because actually it is put for the industries, not for the academic institutions. Okay, because industry spends lots of money into it, so they don't want anyone to copycat them and do it. Am I right? So within that's why the government has given 20 years. All the exclusive rights is for you. Whatever the money that you make, it's up to you. Okay. Can you tell me when the company will go for a patent?
When the process is exclusive.
Yeah, when it goes for it, when they will go for a patent, is it after it is approved, or when in the entire drug discovery cycle? It's a preclinical, okay, clinical, and then human studies.
Clinical, before proceeding for clinical studies.
Uh, Before proceeding for the preclinical, they go for it. Okay, then that is called as a new chemical entity. Okay, then how long do you think that it will take for them to complete the remaining process?
10 years.
10 years. Okay, so in the patent of 20 years, 10 years already gone. Okay, they have only remaining 10 years. In the 10 years, they need to make the money as much as they can get. Okay, so for example, if you look at it during COVID-19 time, the Pfizer vaccine, for every 1 minute, they have earned 2,500 US dollars for every minute. So in returns also that much, and the industries also collapse like that, because they put all this 1.5 billion or 2.5 billion US dollars, because the people who knows about the drug discovery, it is nothing but a gambling. It's a completely gambling game. If you are successful, it's okay. If you are not successful, you are gone. That's why nowadays you can see that majority of the pharmaceutical companies are big pharmaceutical companies, because one of our biggest pride is Ranbaxy. Do you think that Ranbaxy is still there? Yeah. One of the prestigious pharma company of Indian origin. Now it's no more. Okay. So I don't want to tell all those stories, but what I say is drug discovery is not that much jokey. So yeah, can explore covalent overhead. Okay, next one. Then EGFR test study, you open the EGFR target, then you can put the tyrosine kinase inhibitors, you filter the activities, you record the assay data, then you compare the mechanisms, then you can export and carry forward. The output should be a reference set, not just uh, for isolated drug names. So this is the reference molecules and uh, what you need to record. Next one. Okay, what are the common mistakes that they do is copying the best IC50. Suppose if we ask the compound, can you determine what is the most important compound? They compare the IC50 values, and whichever is having a less IC50 value, they say that is the most potent. No, you cannot do that. Actually, you need to check the
సార్ మాట్లాడితే ఆన్లైన్లోకి వెళ్ళిపోతుంది. సార్ సార్ కనిపించు. అది ఆన్ అయితే స్టార్ట్ చేస్తాను. ఇన్ పీపీటీ సార్, పీడిఎఫ్. ఓకే, వెబ్సైట్.
Go to the website. Okay, this is the landing place, okay, where you can do it, okay. Here you can say that ebi.ac.uk, okay, EBI means European Bioinformatics Institute, okay, and of course AC means academic and UK means UK. And madam, could you please go down? These are the things. So far, it has about 2.9 million compounds. Okay, it has about 2.9 million compounds. So there is about 101,000 documents. Okay, and there is a data on 2,200 cell lines. There is data on 18,600 different targets. There is talking about, in total, there is about 7,100 drug mechanisms. In total, there are about 16,100 drug-like molecules. Okay. Then it is also having a 1,700 warnings for the existing medicines. And also there is about 52,200 diseases to which the medicines or drugs are associated. Actually here, you can check this target or you can check from anywhere. Actually, you can move all the things from anywhere to where. Okay, you can check from anywhere to anywhere. Okay, so, and madam, could you please go this one? Okay, of course, in Campbell also, madam, can you click this one, please? Okay, here, actually, what we do is actually this downloads. Actually, what we do is since I'm the subscriber of it, and I can download the entire data into my computer, and I can use this. Actually, from this one, actually, all these are the files that you can download, and you can play with it. Okay, and they have the different versions, and you can download. Okay, if you have a very powerful computer, or otherwise you need to do this. Okay, now okay, go. Here you always need to look at what is the version that we have, Campbell 37. So it is updated only on the 1st May, okay, 2006. And madam, can you go to that again, please? Yeah, where we see downloads. No, you go to the more. Here, don't click that, madam. Here you have a training material. There is a lot of training materials are available where you can get all the things about it. Okay, very fantastic video ones, am I right? And of course, once upon a time in our region, I have my own version of the training material only for Campbell. Okay, then actually it gives you lots of information. Okay, then now we go into directly to here. I will tell you some of the very few things only I will tell you because it contains lots of information. Madam, here you can put EGFR. Uh, okay, EGFR tyrosine kinase targeted compounds, EGFR targeted pro-drug activation. What you can do is you can click this. Okay. Ours is easy. Our target is EGFR only. Right? Okay, you click this one. Anyone you click this one. Or you put simply search also. Okay. Okay, go down, go down, go down. Okay, here, actually here you can see that the journals, European Journal of Medicinal Chemistry, Bioorganic Medicinal Chemistry, European Journal of Medicinal Chemistry, you can see what is the target similarity and all these things, am I right? You can check all the things there. Okay. Now I have told you that whenever you want to compare the activity of the compound, because at the beginning itself what I told you is Campbell is a manually curated database. Okay, that means whenever the paper is there, they will hire some people. Okay, and those people will be paid, and they need to identify the molecule name or the activity name and all those things. Okay, I'm taking only this example. Madam, can you click this one? Yeah, here it is talking about synthesis and anti-cancer effect of some novel pyrimidine derivatives by generating reactants in human breast adenoma carcinoma cells. But actually, we are talking about lung cancer, but not about this one. So it may not be relevant, but you look at the example. Can you go down? Go down. So, okay. Then you click this compound summaries? Yeah. Here, actually, you can look at these are the molecular weights of the compounds, and if this is not mistaken, it should be the log p values of the compounds. Okay, can you go down, madam? Can you go down? Okay, okay. Can you go to this activity charts? Okay, here this is the protein target summary. This is the functional, and this is the bioactive summary. So can you click this one, functional? Okay. Okay, then you are getting all these things here. They talk about the description of the method, and here also there is a description of the method. Okay, so then here they're talking about essay type. This F stands for functional. F stands for functional, and you see these are the compounds. So in total there are 7 compounds which are reported in the scientific literature. Okay, and which was tested on this particular protocol using Homo sapiens. Madam, can you click this 7? I think something is wrong because the alpha fold is also not working. Okay, maybe they might simply put you type any compound name aerotatic or what? ERO, ERO. You can type anything. ERO, TIM. Aerotatic. Okay. Okay, just put. Don't select anything. Just put that. Set. Yeah. Now here you are getting lots of compounds. Am I right? You are getting lots of compounds. Okay. In total, there are 8 records of there. All right. So actually, to work with this, the most important skill that you should get is Excel, because all this data can be imported into the Excel. Once you import into the Excel, then you can use either Python or R language to analyze the statistical analysis. Okay, but this is only a database. Okay, and you need to download everything. And if you put this one, okay, can you click? Or can you minimize this? Here you can see. Can you little bit down? Here you can see that in out of these many molecules, how many are already approved, how many are in phase 3 trial, how many are in the phase 1 clinical trial, and how many is there in still preclinical. Okay, so can you go to the up this? Okay, here I want to tell you a little bit of maybe up. Sorry. Okay, wait. Here you can see erlotinib hydrochloride is a salt. Okay, can you go there? This is erlotinib. This is where the people will get confused. Okay, whether I need to use this data or I need to use this data to collect the ones. Okay, hydrochloric acid means it's only a salt to convert into a formulation. Amarata to improve the stability or whatever. So these are the one things that the people always will get confused. Okay, so again you have to go and check everything about this. Can you check? Can you click this erlotinib? Yeah. Here it talks about the entire chemistry of it. Maximum phase is already approved. It is approved in 2004. You will get all these things. Can you go down? Okay, you can get different formats. Okay, now in all this you stop. Actually, this one smiles ng or standard ng key. Okay, because a large chemical library, we cannot draw the 3D structures. Okay, and of course, if you draw the 3D structures, the file is going to be so big. Okay, because all the people they do the analysis by importing this entire data into Microsoft Excel. So, suppose if you want to know what AI tool that you should learn in drug discovery, my standard answer is Excel. Without Excel, you cannot do anything. Without Excel, you cannot do anything. Any data analysis, you cannot do it. That is the first thing. Second thing is a Python. So a Python language is the one. Actually, all these databases, they use a Python language, okay, to do that one. It's very simple language, okay, to learn. And the third one is R language. Okay, and if you know these three, okay, these are the fundamental AI tools. Okay, whatever we are talking about is that is the cosmetic side of that particular thing. ChatGPT, how it works, no one knows. Okay, and Gemini, no one knows how it works. Okay, in a drug discovery project, you need to have your own database, and if you know that your own database, then you have to use Excel. Okay, I think very recently also I told in one of the talks. Okay, now everyone sees that. Everyone sees that. Oh, students say that, Daddy, I want a computer. Okay, fantastic. What computer you want? I want the best, most powerful computer. Yes. Okay, they will give. So what for they use? They use for the like a stenotyping. Only MS Word only they use. Or otherwise, what for they use? They use games, movies. That's all. And no one even dare to open the Excel. No one even dare to open the Excel. So they buy computer only for the purpose of doing a typewriting. I tell you the Excel, you know, you can in one single sheet, you can enter 7 million points. 7 million data points you can enter in Excel. Okay. Unfortunately, what happens is the people don't know the value of the Excel. Okay. So there, all these chemicals, this thing, we'll use the smiles. That's where we will put structures. And automatically, when you put this one, automatically it will be converted into a 3D structure or 2D structure anywhere. Can you go to this one? Here you can see that there is a copy to clipboard and all those things. Okay. Yeah. Here there is also one thing here I want to show this because there are so many things. This is another powerful one, Shure Campbell. This is the brother of the Campbell, or not, I cannot say the brother, actually it is the son of the Campbell, and which is the most powerful tool. And those who want to go for the patents, this is where they need to, but this one is not free, that means you need to put that. Okay, can you go to this? Yeah. So these are the various forms of this derivative. Okay, this is also available in this form. This is also available in hydrochloride form. Can you go to? Okay. Then, of course, when you look at this molecular features, here it is talking about the Lipinski rule of 5. Madam, click this one. We can click it. Yeah, click it. Yeah, it says that rule of 5 is yes. Can next go to the next one? Molecule, it's a small molecule. Of course, first in class, definitely it is not. There is a chirality. Yes, but there is no chirality at all. Is it a prodrug? It's not a prodrug. Okay. Yes, is it available orally? Yes. Then next one, whether it is available in injection form, it's not. Can you go to this one? Whether it is available in a cream form, it's not. Then go to this one. Whether there are anything mysteries involved into it, no. Then go to this one, availability, prescription only. That means you cannot go and buy in the market. There should be a doctor should write it. Next one is veterinary, whether it can be used for the animal medicine use. No. Can you go to the first one? First one. Yeah. Is it a natural product? It says that it's a natural product. Okay. But we can't even imagine that it's a natural product. Okay. So that kind of information you will get. Can you go to this one? And you can see that this one is useful for the pancreatic cancer, non-small cell lung cancer, esophageal, and all those things. Okay, but can you go up? Yeah. Now, when you go to this one, okay, then here you can go to this. If you want to know about the clinical trials, you put the clinical trials here. Yeah, directly it goes to the clinical trials TOB page. Okay, then it will get all the details. Okay, so yeah, yeah, we can do it. Because from this one you can get all these kind of patents. Okay, now you can see this terminated, completed, and all these things. So now here the thing is, am I right? We talk about AI. Even for this data, am I right? If you want to analyze on your own, you will get lots of headache. Okay, but at this point of time, you must use AI to analyze for you. Again, the same question. Your question must be stronger. Okay, and you need to give this feed because this is the scientifically validated data. You are not plucking somewhere in the world. Right? Of course, I use another one which is called as a NIME. Okay, NIME is called as a code-free machine learning platform. Okay, in the entire NIME, actually I can put whatever I discuss, whatever we discuss. Okay, everything I can put. I can put gene cards. I can put string. I can put what is that Campbell. I can put PubChem. I can put everything, and I can connect, and I can ask what is my expected output. Okay, it will tell you. Okay, so then you have to, what I mean is you have to analyze the data using AI. Okay, so of course Campbell, because usually when we attend the courses or when we conduct the courses, Campbell itself is a whole week program, entire 5-day week program. But you explore as much as you can, and please remember that if you really want, because you are still young, if you really want to master your AI skills, go learn Microsoft Excel first until advanced level. Okay. Then go for a Python and R. And among these two, actually both serve the same purpose. But R is considered as a bit lower version, whereas Python is bit advanced version. Actually, if you know the Python, am I right? And if you are in a drug discovery, actually you can earn lots of millions of dollars. Yeah. Okay. But don't to depend on this all this Gemini, all those things are Claude and all those things. Okay. They are simply using Python. They are simply using Python. Okay, that's why if you ask very complex question, they will give you the Python script. Okay, everything is Python. Okay, so I think that's one. I think we have to stop here. And well, we go to the next one. That is a PubChem, right? PubChem. So actually, we missed one thing. RCSP PDB. Anyway, maybe tomorrow we'll cover that one. RCSP is from UK based one. It is a Royal something Society of Biology or something. Okay. And EBI is European Bioinformatics Institute. Actually, both are situated in London. Okay. But both are the government organizations, but both do the same work. And EBI is the one really fantastic ones. Really fantastic ones. Even AlphaFold is also the European Bioinformatics Institute. Okay. These are the recent introductions. Okay. But before that, this is what is there is a PubChem is there. Okay. But PubChem, after Campbell was introduced, PubChem is not of much use. Okay. It's not of much use in drug discovery. Okay. But PubChem covers more than the drug discovery. More than the drug discovery. Okay. So that is PubChem is mostly everything about chemistry. Okay, and you don't find much information. Of course, they are trying to integrate, but it's not up to that Campbell. Okay, because Campbell was introduced, if I'm not mistaken, should be around 2010 or 2012. Okay, from there onwards, in a matter of 15 years, they have come up with the 37 different versions. Okay, now we look at this PubChem. Okay, set strategy, practical interpretation, common mistakes, and best practices. Okay, this gives you a chemical identity. Okay, and what can you? And for what is that called? You follow that the protocol that was given in your workshop.
Other questions. Detect our lecture. Yes, we detect our.
Okay, can you magnify? Okay, so this is the one. Okay, you can talk about the descriptors, we can talk about the structure stretch, set, and of course the bioassay signals, but it is not that much actually. Okay. PubChem is often the first place research to check compound identity and properties. It is useful for SMILES, formula, CID, that is some particular idea of that particular compound, synonyms, safety summaries, and bioactivity links. The most important thing is it gives only the links. It does not talk about this one. Good PubChem use prevents downstream errors in similarity such alienability and docking, but Campbell also do the same thing. Use PubChem to retrieve the information about this one, and of course you can copy the canonical smiles. Okay, and this PubChem is actually from the American Chemical Society, so you can see chemistry wise it's much stronger, because in the chemistry wise ACS is the pioneer. ACS is the pioneer, American Chemical Society. And PubChem is not just a chemical dictionary, it is a chemical evidence hub for early discovery. The next one, madam. Okay. And this is, madam, next one, previous. Okay. This is a free chemistry source connecting structures, properties, not much of the bioactivity. Okay. But they are trying to add because they are competing with the what is this called as a Campbell. Okay, and you will get a lot of compound records. And if you want to know something about the chemical properties of a particular chemical compound, then PubChem is better. But if you are linking the bioactivity data, and if you are talking about drugs and all those things, then what is it called as Campbell is the good one. Then establishes exact chemical identity, provides inputs for downstream web tools. Okay, here, what inputs it will provide you? It will provide you the smiles. It will provide you the smiles. How many of you have heard about the smiles before this lecture? Okay. How effective do you think that if I give the structure, how many of you know if I give the structure, can you run? How many of you can write the smiles for it? Without using a software help. Without using a software help. Okay. Because that kind of a knowledge a little bit useful. Because of course nowadays the tempera and all those things actually if you right click you can convert chemical structure to the smiles to this one. But in smiles also there are different types. If I'm not mistaken, two are. One is the canonical smiles, another one is the standardized, and there is one more thing is there. And smiles has some kind of a drawbacks also. Okay, and especially if it is a chiral molecule. Okay, so there's another thing called as a smart, and there is also called as an in-cheeky. Each and every one has its unique feature. Okay, so of course, for me also nowadays, what I see is it is becoming difficult to write the smiles pattern. Okay, because the softwares are there. I will click it, got it. But what is the best practice? Okay, you take the structure. Okay, PubChem, it will come to the smiles. You copy the smiles file. Okay, that string. Go to the Kendra or any chemistry software, you convert it into a 3D structure structure, then again you compare. Okay, definitely you will see that 5 to 10% errors, not for all. Okay, there are some they cannot make it, they cannot really make it clear, especially in a salt form. How can you write this file pattern? Okay, so but actually there is something called smile. Okay, smile means because by looking at the structure we will cry. When we look at this thing, oh, so simple. Okay, smiling up. Okay, but there is a elaboration for that which I do not remember exactly what that smile stands for. Okay, so This one canonical smiles, okay, and this drug discovery value, okay, it's provide inputs for downstream web tools. Here I say the inputs is the smiles, link chemistries to bioassays, literature, and patents. Okay. And important caution, and some content is deposited by the external sources, and completed descriptors are not experimental data. That's another problem. Drug-like does not mean clinically useful. Okay. Here, anyone who wants to do it, okay, they can deposit into PubChem. Okay. Whereas Campbell is not like that. It has to, we need to take the prior permission before we put the data. So that's why it is called as a curated database. Okay. And retrieve PubChem and canonical smiles, and these are the XLOGP, total polar surface area, or XBD. Okay. And use the same molecular identity across Campbell's similarity, identity, and everything. Okay, use PubChem to answer what exactly is this molecule before asking whether it is active or developed. Okay, next one. And of course, these are the discovery steps, but most of the things may not work or may not work in the PubChem. Okay, then, but the good thing, what the people oppose, before actually the Campbell is introduced in 2011 or 2012, okay, I always used to depend on the PubChem to download the data and to write the structure activity relationships. Okay, but after the Campbell data, after the Campbell database is introduced, now actually many people are not using it. And this safety awareness, yes, you will get better here. Next one. Okay. Use the exact drug name first, okay, and check synonyms. Use identifiers, okay, and tips for ambiguous names. Many compounds have salt, hydrates, mixers, or stereoisomers. Okay, this is one of the problem. And prefer the parent active compound when preparing files for prediction tools. Okay, so that means even though you are taking a hydrochloric acid salt, when you are trying to determine the biological activity data, you have to put it in a native form, not in a hydrochloric acid salt form. Okay, and those who are interested in a drug discovery project, okay, I think Madam who is working on the nanoparticles. Okay, until now, there is no platform for the inorganic compounds. Okay, there is no platform for the inorganic compounds, but magnesium, zinc, what is that called? Lots of elements. Okay, boron, all are very biologically significant elements. Okay, like selenium. Okay, but until now there is no protocol. Until now there is no analytical tool, or there is no computational tool, especially for the preparation of the, for example, manganese oxide nanoparticles. Okay, unless otherwise we do the molecular dynamics. Okay, Desmond. Desmond is the only software which is available, okay, to computationally simulate all kinds of nanoparticles. Okay, and somehow all the things are going towards organic molecules. Okay, that also I will tell you later. Okay, I'm sorry. Desmond, 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 and there is also one more. Maybe I will check. Maybe tomorrow I will check. Okay, and especially if you are looking for a computational tool for nanoparticles, definitely this is out. Whatever I discussed, all those they don't work. Okay, and that is one area that anyone should explore further, and they will become the pioneers in that. Okay, next. Okay, of course here you will, because sometimes it's very, even now actually PubChem I will get very irritated because they will call it as substance. They will also call it as a compound. It's very difficult to identify what is the difference between a compound and a substance. Okay, so here compound means standardized unique chemical structure with CID. Okay, this is the compound ID, CID. Okay, this substance means, suppose, madam, if you give one compound and if you think that you can give your own name, okay, then I do the same thing, I can give my own name. So there is no standardized ones, and I will get very irritated with this one whenever I look at this one. Okay, this is called substance descriptor depositor provided chemical substance record. Of course, we have the bioassays, and also you have the patent or literature links. So I always will get a lots of these things. Of course, when you are looking at also, when you put a particular compound, same chemical structure, lots of what is it called, output will come, lots of things will come from the same compound. So sometimes it is very irritating. Okay. Explore biological, support novelty and background exploration. Okay. Then these are the common mistakes that what we do, using a salt record as the parent drug. That's not at all. Okay. Just I'm asking, what is the purpose of preparing in a salt form?
Solubility.
Only for the solubility.
Stability.
Stability. Then
Bioavailability.
bioavailability. Then
To reduce the toxicity of the
it
drug.
reduces the toxicity. Okay. If that is the case, in drug discovery, some molecule, some people will take this one that is not safe, and they always scratch their head. It is not safe. It is not safe. Sir, it is not soluble. It is not soluble. It is not soluble. And converting it to salt form is easier, am I right? Why they don't do it? One of the biggest challenges in the drug discovery cycle is solubility issue. And most of the compounds that I synthesize, 70 to 80, 60 to 70% of them, they never soluble.
One of the techniques for enhanced solubility, it has limitations like microscopes, sticky issues.
Yeah. So actually, salts forms we try to avoid as much as we can because of the impurities. Okay, because of the impurities, we try to avoid. That's why olden days we used to have lots of salt form drugs, and in the recent days, actually you don't see salt forms. You will see less and less and less. Okay, so use compound for standardized chemical identity and substance for deposited source records. Okay, next one. Then, of course, when you open this one, you will have the 2D structure, 3D structure, synonyms, and formula. Okay, and these are the computed properties. Okay, hydrogen bond donors, hydrogen bond acceptors. Okay, can you tell me, oxygen, is it a hydrogen bond donor or hydrogen bond acceptor? What is OH? Hydrogen bond. Okay, because in the entire drug discovery, there are two elements that are dominating. That is oxygen and nitrogen. Okay, and it's very difficult to determine which is a hydrogen bond donor and which is a hydrogen bond acceptor. It depends upon the predominant form of that either oxygen or nitrogen. Okay, among this one, the classical example is amino group. Okay, Amino group is supposed to be what? Hydrogen bond donors. Am I right? Hydrogen. But however, in a biological system, in the acidic environment, okay, it will form NH3 plus. Then it will change completely its properties. Okay, so that's why the oxygen chemistry and nitrogen chemistry is something which is very difficult to understand, which is very difficult to understand how they behave. Of course, in the standard organic chemistry textbooks, simply will say that this is hydrogen bond donor, this is hydrogen bond acceptor, but no one really talks about the predominant form of. How many of you studied the Henderson-Hasselbalch equation? Okay, that is the main thing that rules the entire drug discovery. Because Henderson-Hasselbalch equation, whenever we study, we see that, oh yeah, this is very boring works. Okay, it talks about the ionization, unionization, percentage ionized, percent ionized. Okay, but if you want to determine whether the molecule is going to be hydrogen bond donor or hydrogen bond acceptor, you need to know the Henderson equation. Because in Henderson equation, what it says is a particular molecule can exist in a different forms in different pH environments. Am I right? So when you are changing the pH, at one pH it may be in more ionic form, whereas in another pH it will be in more in unionized form. If you look at our biological system, we have all types of pH. We have the acidic pH in the stomach. When it goes to the blood, it is a neutral, and when it goes to the further part down, small intestine, it is a basic. So then you imagine, you take a same molecule, it will pass through all these three compartments. Then it will exist in a different forms. That's why drug discovery is very difficult. Okay, hydrogen bond donors and hydrogen bond acceptors, whatever we see, it's not so easy. Because, and most of the time, we will forget about this fundamental equation, Henderson-Hasselbalch equation. And that is the fundamental ones in drug discovery, especially in designing the chemical compounds. Okay, and so yeah, and of course the total polar surface area, this x log p. Okay, of course I think today is my last one is talking about x log p. Why it should be in a log form? And what is meant by x log p? How many of you heard about the apparent partition coefficient? Apparent partition coefficient. Okay, now it's a very simple. Most of the textbooks, what they say is partition coefficient is the amount of concentration of the compound in the organic phase over the concentration of the compound in the aqueous phase. Okay, but they never talk about what is the ionized form and unionized form. That's where the apparent partition coefficient will come. Okay, because if you take a particular bond, if you take a particular molecule, okay, suppose you take organic phase versus aqueous phase. Sorry, organic phase versus aqueous phase. Okay, you know that for the compound to be soluble in an aqueous solvent, it should be in an ionized form. Am I right? It should be in an ionized form. So if you take a particular drug, it is going through the different pH compartments in our body. So in one compartment, it may be soluble in water, but in another compartment, it may not be that much soluble in water. Am I right? Because the ionized component will be less based upon the drug. Okay, that's what called as apparent partition coefficient. That's why you imagine when you take a drug, you are putting in a water and you are drinking it. It is going into your stomach or what is gastrointestinal tract. It should be soluble in water. Am I right? So it should be in more ionized form. Am I right? Then it has to cross that. What is that called? Lipid bilayer into the blood. Then, if it is completely water soluble, how it can cross through? Again, it will change. Then it will go inside. Once it goes inside, again it has to be in ionized form. Then only it will be soluble in blood or plasma. Then it goes to the different compartments. When it goes to the different compartments, it has to go into the different tissues. In order to go into the different tissues, again, it should be in an unionized form. Okay, that's why a molecule will change its properties throughout its journey. Okay, until it comes, when ultimately whatever we take, it has to come out from the urine. Then again, it has to be soluble in water. Okay, that's why designing a chemical compound is not that much easy without understanding what is meant by Henderson-Hasselbalch equation in drug discovery, without understanding what is meant by the apparent partition coefficient, and without understanding what are the various pHs values in our body. Okay, that's the one of the biggest. That's why the X log P value is there. Okay, it's not a simple partition coefficient. Okay, and also the rotatable bonds and complexity. Okay, smiles, inchi and inchiki. Okay, and coordinate and file downloads. Okay, the rotatable bonds. Am I right? Suppose you take these things, all these are rotatable. Am I right? Something rotatable. Okay, since these are rotatable, what happens is you can eat properly. You restrict one. Do you think that you can eat? Am I right? So, but at the same time, it is true when you are becoming old, what happens is still there's so much rotatable we cannot even control. Even you cannot take the food. So, in drug, what is it called? In the case of a drug discovery, molecule should be rotatable. That means before it is going into the receptor, it should fold, it should go inside, and it should be again open up. Okay, but at the same time, it is not should be too much rotatable. Okay, that's why the number of rotatable bonds they will say maximum should not be more than 10. Okay, because what I'm trying to say here is on paper everything looks nice. Okay, and most of the people they design the molecules by only looking at the structure without understanding how it behaves within the biological system. Okay, that's the very important thing. That's the reason why I say that the pharmacy is the best program, because they come to know about all these things together, and if they do it properly, they can connect all these points in a better way. Okay. So then, of course, linked evidence. Do not try to read the whole page in one sitting. Actually, this is applicable for everything. Okay. Then decide whether you need deeper evidence. Okay. Next. Madam, 3 minutes only. Just okay. Sorry. Okay. Then why the smiles matter? Smiles is a text representation. Okay. And Swiss similarity and Swiss ADV, you can use smiles directly. A single missing character can produce a wrong molecule. This is one of the things. The people simply copy and paste. Okay. And in that one, you can see there is a capital C, small c is there. And many people do not know what is the difference between the capital C and small c. Okay. Sometimes when they paste it wrongly, they will get it a completely wrong, completely wrong. If you change small c to the capital C, that's all. You will get everything wrong. Okay? So copy from the canonical smiles field, not from memory. Okay. This is one. Paste into a plain text file before using other tools. Okay. Let us say this is what I say. We have the Microsoft Word, we have the Notepad, we have the WordPad. Okay. In all these things, actually, we can enter the data. Okay. But if you are preparing a document for office work, you can use MS Word. But if you want to use for drug discovery, everything you have to use Notepad, not either WordPad or Microsoft Word. You must use Notepad. Okay. Why? Of course, there are lots of things to read. I don't want to. Okay, that's why they say that paste into the plain text file, keep one molecule per line and label each line clearly. Okay. So next one. Okay, this is molecular weight, Hex log P, HPD, EPSA, rotatable bond. Okay, here high flexibility can reduce oral bioavailability, but no one knows how much flexible it is. Okay, here, how many of you know about the Lipinski rule? Fantastic, am I right? So, do you think that it is the best one in medicinal chemistry? Yes or no? Students, yes or no? Yes. Can you tell me any antibiotic which obeys the Lipinski rule of 5? Have you seen the structures of the antibiotics? How big they are? Then can you tell me Lipinski is really good? This is simply the misinterpretation of the present generation. Lipinski rule of 5 is not a rule. Okay, Lipinski is a person who worked in a Pfizer company. So he is the head of the research wing at the time. So that means in early 2000s. Okay, so early 2000s. So he analyzed all the molecules that are converted into medicines. Okay, then he come up with this Lipinski rule of 5. So the database that is available at that point of time, he said that 95% of the compounds obey these 5. Because Lipinski rule of 5, because it's a bit of fancy term, and the people started thinking that, oh, drug discovery means Lipinski rule of 5 is the thumb rule. Actually, it's not. If anyone follows the Lipinski rule of 5, they never invent anything in their lifetime. Okay. So because what I need is, because it's the way the people talk. Okay. That's why the people have misinterpreted the Lipinski rule of 5. Actually, it's not the rule. It's a simple observation that what they have. Okay. Of course, this is what this one, in what way you will be careful, and this is what about the people are talking about. Okay. And here I will tell you this simple example of. Okay. Next. Okay, you can also use for the identity search, and you also can do this similarity search, and you can also search the substructure search. Okay, all these things you can do it. Choose the search mode based on the scientific question, and it's not a habit. Okay, suppose if you want to find the same structure, that means if you want to identify a single compound, you go for the identity search, and if you want to go for a similarity search, that means if you want to get the molecules which are having a similar physicochemical properties, you can go for this similarity search. Or if you want to go for a substructure search, okay, always we talk about a pharmacophore. Am I right? In a drug molecule, there is some particular component which is good, which is mainly required. Okay, can you tell me what is meant by pharmacophore? What is meant by pharmacophore? Yesterday we have seen the structures, am I right? The first 3 structures, it is very common. Actually, what does this do? Pharmacophore will do what it will do actually. Actually, pharmacophore is the one actually that determines to which receptor it will go and stick in. Yesterday I asked one thing, how does this chemical, even though it does not have brain, how it will go and stick there? Okay, that is the pharmacophore. So if someone wants to look at the substructure search, then they do it. Of course, in our medicinal chemistry textbook, the way they define the pharmacophore is the one that determines the activity and all those things. But strictly speaking, from the drug discovery perspective, it is not exactly the truth. Okay, from drug discovery perspective, pharmacophore is the one that recognizes the receptor where it should go and sit. Okay. Then, okay, next one. Yeah, this is what I tell you, what similarity can tell you, and what it cannot tell you. Okay. Again, the compounds may be similar, but they may not produce similar biological response. Okay. All of you know about isomerism, am I right? Plus and minus, am I right? Can anyone explain why the plus is active, minus is inactive? There are lots of drugs that are available in the market. Only plus isomer is active, whereas minus isomer is inactive. Entire structure is same. Only the orientation different, am I right? Only the orientation is different. That's why when you draw the structure, how do we identify whether it is going to be upward or whether it is going to be downwards? Am I right? Unfortunately, on a piece of paper, you cannot draw the chemical structure. You cannot draw the chemical structure in 3D format. There you can use AI. Okay, you can ask AI to produce a 3D model of it. Then from that, actually, you can decide whether you want to be attach a particular substituent in alpha orientation or a beta orientation, whatever you want. Okay, that's the next. So then you look at what the bio essay contains. Then you need to read how to entire essay description, and then some people they call it as active without reading essay context. That's very important. Okay? And use bioessay to generate leads for develop deeper validation, then verify with the curated bioactivity sources such as Campbell. Here again, this particular statement is very, very important. They say that even PubChem also say that go and check with Campbell. Okay. Next one. Yeah, of course there is a hazard pictograms. This is toxicity. Actually, for example, okay, because there are some people with the nanoparticles. Okay. You know, there is one concept. There is a one concept that what the people talk about. Okay. Somehow, in some parts of the world, nano is not well accepted. Okay. So if you look at most of the papers, majority of the papers come from the Asian countries. I'm talking about from the drug discovery point. I'm not talking about nano is fantastic in all these things. Okay, that is really fantastic. There is no doubt about it. Only I'm talking about a drug discovery. The thing is, nano means small particle, am I right? So it can go very easily. It can go into very good, therefore toxicity is going to be much, much higher. But most of the time I say that nanoparticle targeted drug delivery system, many people talk about it. Okay. And a lot of the pharmaceutics people, they do lots of research on nanoparticle targeted drug delivery system. Can you tell me a single medicine which is available in a nano form? Of course, now for anti-cancer drugs it is there. Okay. So because without knowing, in one of the conferences in Europe, I talked a lot about it. Then I got lots of scoldings from them. Then I need to realize that nano is nano. You can produce a nano car, but don't try to produce nano medicine and all those things. It's not so useful, okay, to be honest. Okay, and so this is the drug discovery that what we do. Okay, next one. Yeah, this is the question. This is the PubChem strength. This is the Campbell strength. Actually, this is where you can compare the PubChem versus Campbell. Okay, next one. And these are the mistakes that what we do. Okay, we use the wrong salt, copying the smiles. Okay, and treating compound properties as the experimental truth, but it's not. Okay, assuming PubChem bioassay active like a drug candidate. Sometimes we miss the stereochemistry. These are the best practices that you always need to look at it. Okay, the main PubChem skill is not clicking. Okay, it is knowing what not to overclaim. Actually, this particular statement is applicable for all the databases. Don't overclaim anything. Okay, next. Of course, this is the step-by-step method. And save the PubChem CID and URL. Okay, in your worksheet. Record whether the molecule is a parent compound, salt, or this one. Keep search mode and threshold if using structure similarity 1. Write one sentence explaining why the compound was selected. Okay, okay. Madam, can you go this? So you say this is one. Instead of writing PubChem showed the compound is good, but instead of that you got to write PubChem so and so CID was used to retrieve the chemical identity, canonical smiles, and computer descriptors for downstream ADME prediction. That's what you need to say. Next one. Yeah, these are the weak wording, and this is the better wording, and why it is better. This one. Okay. A good method statements allows other researchers to retrieve the same compound record and repeat the analysis. Okay. Okay. You know, when we are writing a paper, we write an experimental method. Am I right? Do you know what is the main purpose of writing an experimental method? Whether in a thesis we write so much, am I right? Whereas in paper only we write this much. Actually, what is the concept? What is the concept? Yeah. Actually, if you look at all the journal guidelines, what they say is the experimental protocol that you write in a paper should be concise, should be understandable by other researchers, and should be reproducible. That's the most important thing. And many people, they do not know, oh, I have this much of experimental method, I have to concise it. Without knowing this one, they will concise it, and later on they say that, oh, my paper got rejected. Okay, that's one thing. So actually, yeah. Next one. Yeah, of course, this participant talks and all those we can do it. And later on, tomorrow we are going to talk about Swiss similarity and Swiss ADAB, which talks about it. Okay. Then next. Yeah. Of course, this is manual download. Okay. And this batch workflows are programmatic access. Okay. And actually, what happens is this PubChem database is linked to whatever the workflows that you generate, then automatically it will be curated. Actually, you can do it even PubMed also. Like the morning you asked me about RSS alert and all those things. Even PubChem also you can do it, especially for the nanoparticles. For engineering people, I think you are from engineering. Yeah, chemistry. But most of the engineering people, they would still work on the PubChem. Okay, when they want to choose manganese oxide versus zinc oxide versus silver, or what nanoparticle we should use. Okay, and most of the people they use PubChem. Thank you. Yeah. Oh, still there. How many more? I think that's the end. Can you go to this? Yeah. Yeah, here the final best practice workflow, how I would use PubChem in the early EGFR drug discoveries. This is the stage that you want to do it. Here you can use the PubChem. Okay, when you want to compare the descriptors and structure similarity and everything, and this is one thing that you will get it. Okay, that's the okay. That is about PubChem. So tomorrow we are going to talk about Swiss similarity, Swiss ADME. I will find out RCSP PDB, and there is one talk that I prepared. What are the various tools that we can use in drug discovery? And I also have written some pros and cons about it, if I'm not mistaken. That is going to be the last one. Okay, so with that we are going to. Okay, sorry, madam, I took about.
Thank you, sir. Thank you very much for a wonderful session. With this, we end the day two session of a 3-day hands-on workshop on artificial intelligence in drug discovery and development in lung cancer case study. So, with this, we conclude this today's session, and we'll meet tomorrow at 9:30 a.m. Thank you, everyone. Thank you for participating in this session.